Well, hello there and welcome back to our series of videos on the topic of inheritance. Now in this video, we're going to continue looking at different patterns of inheritance, and in particular, we're going to take a look at how blood type is inherited. We're going to see how blood type is an example of codominance, as well as multiple alleles. And this is very important for your syllabus. So to begin with, we know that there are 4 different blood groups in humans: A blood type, B blood type, O blood type, and AB blood type. So hopefully you can recall that there's different antigens present on the red blood cells of each blood group. So for A blood group, the red blood cells have A antigen on their surface. For B blood group, the red blood cells have B antigens. And for AB blood group, the red blood cells have both A and B antigens. And for O blood group, the red blood cells do not have any antigen, do not have A or B antigens on their surface. So it turns out that the gene that controls blood group has what we call multiple alleles. It exists in more than 2 forms. Now this is different from the other examples we've seen so far. You know, when we looked at Mendel and the tall and short pea plants, we saw that the gene for height existed in 2 forms: the capital T, tall form, or the small t, short form. But in this case, blood group doesn't just have 2 alleles, right? The gene that controls blood group actually has 3 possible alleles. So we say that it has multiple alleles. It comes in 3 forms. The first form is the IA form, and this allele codes for the A antigen. So if you have this allele, then you have the A antigen. The second form is called the IB allele. IB allele codes for the B antigen. And the last form is the IO allele. So the IO allele does not code for any antigen. You might be wondering what this I stands for. This I stands for immunoglobulin. Immunoglobulin. Which is just a very complicated way of referring to the antigen. So as per all the examples we've seen so far, each individual has a pair of alleles for blood group. So each individual has only 2 alleles for blood group. So here are the possible genotypes for blood group, and I want you to think about what are the phenotypes in each case. So an individual may have the genotype IAIA, and in this case, his phenotype would be A blood group, right? Because he only has the IA allele. An individual with genotype IBIB would be B blood group, and genotype IOIO would be O blood group. Now, how about the genotype IAIO? Well, this person here will have the allele for A antigens, and this one doesn't produce any antigen. So actually, his phenotype at the end of the day would be A, blood group A. And if a person has a genotype IAIB, then his phenotype would be AB, because he has both the A and B antigens. And finally, the genotype IBIO would be B blood type. So in this case, what do you notice? Which allele is dominant, which allele is recessive, and are there any surprises? So it turns out that we can say that both the IA and IB allele are dominant over IO. Because when there's a genotype IAIO, the resulting phenotype would be just A blood type, which is the phenotype of the IA allele. So that means that IA is dominant over IO. The same thing for IB, because when there's a genotype IBIO, then the phenotype will be blood type B. So IB allele is dominant over IO. And then you can say that IA and IB alleles exhibit codominance. Now what's codominance? Codominance is a situation where both alleles controlling a trait are distinctly expressed in the phenotype. So when the genotype is IAIB, then we saw that the phenotype is an AB blood group. So in this case, you can see that both alleles, the IA and the IB allele, both alleles are distinctly expressed in the phenotype. They're both present in the phenotype. So when this happens, then we say that it's a case of codominance. So with this in mind, it's also helpful to know what are the possible genotypes for each phenotype. So if a person is type A, blood type A, he actually has two possible genotypes. He could be either IAIA or IAIO. If he's blood type B, he could be either IBIB or IBIO. But if a person is blood type AB, he can only be genotype IAIB. And if a person is blood type O, he can only be genotype IOIO. So if we see a person with blood type AB or O, we can be fairly certain of their genotype. But if we see a person of blood type A or blood type B, then we need to do a bit more investigation. We can look at their children, or we can look at their parents to kind of determine whether or not which genotype they are. So there's a practice example for us to try before we finish up this topic. In this example, we see that a woman with blood type O married a man with blood type A. And the first part asks you to explain how it's possible for the couple to have a child with blood group O. So in such questions, it's always very helpful to ask ourselves, what can we be certain of? So the first thing I can be certain of is because this woman is blood group O, we know that definitely her genotype is IOIO. And if they have a child of blood group O, then we know that the child's genotype is also IOIO. Now, however, this man is blood group A, and blood group A, you can either be IAIA or IAIO. So which one is it? Well, in this case, because the man has an offspring who is IOIO, it means that he must have the IO allele, right? So that he can pass it on to his child. So in this case, his genotype is probably IAIO. And that's how we answer the first part of the question. So if the mother is blood type O, IOIO, and the father is IAIO, then we use the Punnett square, we can actually see the resulting offspring. So here's the mother's gametes, IO or IO, and here's the father's gametes, IAIO. And so the possible combinations, we'll see that half of their offspring will be blood type A, while the other half will be blood type O. So it's possible for a mom of type O and a father of type A to have offspring who are type O. Now the second part of the question asks you, what is the chance of your first child having blood group A? Assuming that the father is heterozygous. So if the father is heterozygous, means that he's IAIO, right? Because it means that he has different alleles in his genotype. And the third part asks you, assume that the father is heterozygous again. His first child is blood group A, and the father thinks that his next child will be blood group O. What's the chances that it will be correct? So if we take a look at our genetic, our Punnett square, according to our Punnett square, there's a 50% chance of blood type A. So there's a 50% chance that his child will be blood type A. And his next child will have a 50% chance of being blood type O. So just because his first child is blood type A doesn't mean that his next one will definitely be blood type O. Every child is an independent event, so the probability will be the same for every child. Okay, so there's a checkpoint question for you to try. And in this question, it's asking you, what would be the genotype of person individual number 1? So what is this person's genotype? So how do you go about this kind of question? Well, the first thing you have to say is that because person 1 is blood type A, right? Because he's blood type A, so his genotype is either IAIA or IOIA, right? However, what clues we have? So we know it's either IAIA or IOIA. So we have to look at either his offspring or his parents. So in this case, we look at his offspring. And we notice that one of his offspring is an O blood type, right? So one of offspring, person number 6, is blood type O, which means that it's definitely genotype IOIO. So what does that mean? That means that person number 1's genotype is definitely IOIA, right? So therefore, person A is IAIO, so that he can pass on the IO allele to person 6, who is his offspring. Right? Otherwise, his offspring will not be blood type O. So that's how you do this kind of question. Alright, finally, we're going to take a look at traits with continuous variation. So for traits with continuous variation, first you need to remember what's continuous variation. So recall that continuous variation refers to traits that have a range of possible phenotypes between two extremes. So continuous variation means that I have many possible range of phenotypes. So it could be very light or very dark skin, for example. Right? Or we looked at examples such as height, so you can be very short or very tall, weight very light or very heavy. Right? What's important is that there's not just two clear-cut categories, not just a few clear-cut categories, but a range of phenotypes. So a question that comes out is, how are traits with continuous variation controlled? And it turns out that such traits are controlled by many genes, not just one gene position on a chromosome. So it's actually controlled by many gene loci. And continuous variation, traits with continuous variation is actually due to the additive effect of many gene loci or many gene positions. So let me show you an example in the next slide. And it is this additive effect that produces that range in phenotypes. So I want you to imagine that there are 3 gene loci for skin color. So for example, I might have, this is my chromosome, this is my chromosome, and there's 3 gene loci. So this is gene loci 1, gene loci 2, and gene loci 3. 1, 2, 3. So actually there's 6 alleles in all, right? So there's one pair of alleles here, another pair here, and another pair here. Now I want you to imagine that these genes can either be a dark form or a light form. Okay, so each individual has 6 of these alleles. 3 from your mom and 3 from your dad. And they can either come in dark or light form. So imagine if your mom gave you 3 copies of the dark allele and your dad gave you 3 copies of the light allele. Right? Then in total you'll have 3 dark and 3 light. So your skin color will be somewhere in the middle. But imagine if your mom gave you 3 dark and your dad also gave you 3 dark. Then in total you'll have 6 dark alleles and you'll be super dark. Right? So it's because there are many gene loci that there's actually a range of phenotypes. And the skin color at the end of the day depends on the total number of, for example, dark alleles that you have. Alright, so here's a very complicated way of showing it. I use different letters to represent each gene loci. So letter A represents the first loci, letter B and then letter C represents the third loci, and so on. Okay, this is very complicated, so I'm just going to skip over it. What's important is on the next slide. So what's most important is that you know how to compare between discontinuous variation and continuous variation. So in the case of discontinuous variation, remember that it refers to a few clear categories of phenotypes, for example, tall versus short pea plants. For continuous variation, it's a range of phenotypes. And for discontinuous variation, it's usually controlled by only one gene. Right? We've only looked at examples with only controlled by one gene, right? So the height gene, which only came in 2 forms, big T or small t. But for skin color, it may be controlled by many gene loci, many gene positions. In discontinuous variation, because it is only controlled by one gene, or at most maybe 2 genes, the genes do not have an additive effect. But in continuous variation, the genes have an additive effect. So just now you saw that even though there are 3 gene positions, all of those gene positions will contribute to the skin color. So they all add up together to contribute to the final skin color. And finally, discontinuous variation is not very affected by the environment, while continuous variation is more affected by the environment. Alright, so that's all for this video, and I'm not going to be covering sex-linked traits, but if you're interested, you can always search online for it. There are many good videos about it online. And it's very interesting because it has things to do with like color blindness and certain males getting a disease more than females, etc. So I'll see you in class. All the best.